Welcome to the Neurostimulation podcast. I'm Michael Passmore, Clinical Associate Professor in the Department of Psychiatry at the University of British Columbia in Vancouver, Canada. The Neurostimulation podcast is all about exploring the world of neuroscience and clinical neurostimulation. We talk about the brain, how it works, the latest research breakthroughs in neurostimulation, and how that research is being translated into real-world treatments that can improve health and well-being. This podcast is separate from my clinical and academic roles and is part of my personal effort to bring neuroscience education to the general public. Accordingly, I would like to emphasize that the information shared in this podcast is for educational purposes only and is not intended as medical advice or a substitute for professional medical guidance. Today's episode is presented by ZipStim Neurostimulation. ZipStim is the neurostimulation clinic that I operate. You can find out more about our clinical programs at zipstim.com. That's zipstim.com. Today, I'm really looking forward to a discussion with Dr. David Blake, Professor of Neuroscience and Regenerative Medicine at Augusta University in Georgia. Dr. Blake's lab is pioneering work in deep brain stimulation and its effects on aging and cognitive function, particularly through the use of cerebrospinal fluid markers in non-human primate models. In this episode, we're going to dig into Dr. Blake's lab's recent findings as they presented at the 2025 Brain Stimulation Conference in Kobe, Japan, and we're going to explore what those findings could mean for the future of neurotherapeutics, particularly in age-related cognitive decline. The title of their poster is a bit of a mouthful, but I'm going to give it a shot. Exploration of deep brain stimulation effects on neurotrophic markers in cerebrospinal fluid in aged rhesus macaque monkeys using an omaya. Dr. Blake's lab in this poster offered the following background as a way of setting the stage for this project. They noted that the losses in cognitive function with aging occur in parallel with decreases in function in the forebrain's cholinergic systems. Now, this is the primary target for the medications that are approved for stabilization of mild to moderate Alzheimer's disease due to the targeting of that exact forebrain cholinergic system. The cognitive impairment is concentrated in executive function and memory loss. And the team noted that prior work applying electrical stimulation to the nucleus basalis of Meinert has documented that intermittent but not continuous stimulation for 1 hour per day improved working memory task performance in adult monkeys. So this team's study tested the impact of intermittent stimulation on behavior in macaque monkeys and control animals either delivered a donepezil intervention or unstimulated. So we're going to have a discussion about this particular study and other relevant work in Dr. Blake's lab. Welcome back to the Neurostimulation podcast. I'm here today with Dr. David Blake, Professor of Neuroscience at Augusta University in Georgia. Dr. Blake, thanks so much for joining us today.
Oh, I'm happy to be here.
Maybe if you don't mind, could you introduce yourself and talk a little bit about your position there, your team's work, and then I'd love to get into some more detailed discussion about the research, in particular, your lab's work that was presented at the recent conference in Japan in February.
Yeah, so my work, my lab's work for most of the last decade is focused on stimulation of the basal forebrain in monkeys or in humans. That would be the nucleus basalis of Meyer. That area has neurons that project to the cerebrocortical mantle, and most of those neurons release acetylcholine. So we think that we're controlling or have some control over the acetylcholine tone in the cerebral cortex by virtue of our stimulation. And the observations that we made suggest that we can use it to improve executive function in a wide variety of mammals, mice, rats, monkeys, and humans. And so we're both studying the process and also pushing into getting it into the clinical realm. My
Yeah.
background is in biomedical engineering, trained in neurophysiology. My graduate work was at Johns Hopkins studying sensory processes, and I studied sensory cortex plasticity at UCSF for a decade. And then serendipitously backed into a corner of neurostimulation, and then it's all been taking off from there.
Fantastic. Yeah, it's interesting how things go in terms of when one gets through the training and then you get interested in one direction or another, and then these interests and opportunities coalesce. So that's been, yeah, great to hear about how that all evolved for you. I
In
was,
fact, for a little segue, I tell all of our trainees that if you observe something that's just seems too fantastic and is not really related to your primary scientific question, it's often worth investigating a little bit more because it can really become a launching pad for a new scientific question. You shouldn't just ignore it if it's not suiting your immediate goal.
yeah, for sure. No, I think that would be an interesting part of the discussion too, is talking a bit about things like mentorship and encouraging trainees. So this study in particular, I was looking back through and reviewing the poster, exploration of deep brain stimulation effects on neurotrophic markers in cerebrospinal fluid in aged rhesus macaque monkeys using an Omaya. So maybe do you mind helping us to understand a bit about that study in particular?
Yeah, so this was the newest thing that was added to the paper that was published in Brain Stimulation this past January. So people can go look up that paper and see the fine details and study them at as much detail as they like. We got interested in deep brain stimulation because it caused plasticity in sensory cortex, and then started studying what happens if we stimulate while the animal's doing a behavior. And then we switched to studying executive function behaviors and found that we could cause enduring improvements in executive function behaviors. Now, we were really interested in how this all worked. And being an electrophysiologist, we did some recordings. I worked on this, a lot of this jointly with Christos Constantinidis at Vanderbilt. And the electrophysiological correlates that related to the magnitude of the cognitive effects just weren't there. We would apply the deep brain stimulation. We'd see very subtle effects happening in neural responses. It just, something else had to be happening that we couldn't really see. And we did a mouse study that we published in Cerebral Cortex a few years ago, where we could do protein expression and look and see what's really changing in the neural tissue. And the big hits that came out of that were the receptors for nerve growth factor and brain-derived neurotrophic factor. Now, those are neurotrophic receptors. So if they're activated, they cause the part of the cell on which they're expressed to grow. And these cells are growing. Well, what activates them? Nerve growth factor is part of the cholinergic signaling pathway in the forebrain and in the cerebral cortex. So tissue that produces nerve growth factor draws cholinergic axons in and causes them to release more acetylcholine. So we were excited to see that. We were a little wondering why we saw the BDNF at the same time, brain-derived neurotrophic factor, which doesn't relate so directly to the cholinergic pathways that we were stimulating. But both of those neurotrophic factors are activated in 6-hour-long processes where the nerve growth factor peptide binds to the receptor, it gets internalized, it gets phosphorylated, transported back to the nucleus, changes gene and protein expression in the cell, which causes that part of the cell to grow. So it's not something you could see electrophysiologically. So we were wondering, how can we see the immediate results of this process? And we started sampling cerebrospinal fluid. And of course, the way you typically do that is through a spinal tap. And that's what we did to get our initial data. But I worked on this collaboratively with the neurosurgical team. The chair of neurosurgery is very interested. He's a functional neurosurgeon, which means he does deep brain stimulation clinically. And so he's interested in new applications of deep brain stimulation. So he came in and started doing our monkey surgeries with us, which, by the way, is fantastic if you have a neurosurgeon helping you with your monkey surgeries, because they get done better and you learn a lot more about your surgical skills. He said we can try using this Omaya. Now, for those who are not neurologists or neurosurgeons, an Omaya is an extraventricular drain and reservoir. So it's a little tube that goes into the ventricular system of the brain that's about 2 or 3 millimeters wide. And the tube goes through a small hole in the cranium to a reservoir that's under the skin. And the whole system doesn't hold very much fluid. It holds about 50 microliters. But you can stick a needle into it and allow the positive pressure of the CSF to allow cerebrospinal fluid to flow out. So you can sample cerebrospinal fluid. Now, this is really interesting. And there's been tons of studies on the neurotrophic receptors and the neurotrophins in rodents and in culture. But you can't do the cerebrospinal fluid studies in small mammal models because you can't sample large enough volumes of cerebrospinal fluid to be able to measure protein expression. Now, the macaque doesn't have that problem. We could easily sample, and we actually went through studies and how much can we sample over what length of time. And it seemed like a quarter cc could be sampled every 30 minutes, which is about 1/4 of the production of CSF from the macaque, ongoing production. We could sample that every 30 minutes. And so that's more than enough to do really nice protein expression, which is the next phase of the project. So with all those things in place, we took the monkeys we were studying cognitive changes in, and we would stimulate them and then draw cerebrospinal fluid after a half hour, after an hour. And we did the first things we looked at were the pathways we thought should be involved in the cerebrospinal fluid for the neurotrophic receptors. So that's the neurogrowth factor receptor and the brain-derived neurotrophic factor receptor. We should see their peptides, and we should see tissue plasminogen activator. Tissue plasminogen activator is well known for people that study stroke because it's given to ischemic stroke patients if they're caught in the first 6 or 7 hours, and they meet other certain criteria because it breaks the clots up. It does other things to activate biochemical pathways in the cerebrospinal fluid. It activates nerve growth factor, and it activates brain-derived neurotrophic factor. So these were just the first things we wanted to look for, and we found them in spades. And so we're actually really excited about the next phase of study where we'll go in and use a more comprehensive proteomic approach to look at this. We're interested in these 2 neurotrophins and this activator. There's going to be hundreds of proteins that change in their expression levels when we stimulate, and we need to get a more holistic picture of what's going on, which we just can't do looking at one protein at a time.
Yeah. Wow. That's fantastic. It's such an interesting evolution in terms of the inception, the theory behind it, and then these practical matters that you're describing. It's really interesting. It's fascinating how you have these intersections between and the access to experts like with the neurosurgeon as well.
We started to look at the cerebrospinal fluid. We're like, why hasn't anyone else done this? And you can't draw serial samples in rodents and humans that, especially like a subarachnoid hemorrhage patient would have an extraventricular drain placed anyway. People haven't done systematic manipulations and drawn fluid in those cases because probably they're not working in the neuro ICU and thinking about these questions. But it's once you're set up, there's a whole set of really interesting questions about what's happening in cerebrospinal fluid. And we just don't know. For example, if you do an executive function behavior for an hour, you're thinking really hard for an hour. What changes in the cerebrospinal fluid? You don't really know. Or aerobic exercise improves executive function in virtually every mammal that we know of. And there's got to be markers that are changing in the cerebrospinal fluid when aerobic exercise happens. We don't really have a good clue as to what those are either. We have some hypotheses, but the direct assessment hasn't really been done. And this preparation will allow those questions to be addressed directly.
Yeah, for sure. It's really interesting because I think that hopefully that is going to be something, there's obvious real value in getting this kind of detailed data from these tissue sources and the CSF sources, because then that can allow for that analysis. And then as you say, the sampling of the proteins and the analysis of how the genomics perhaps are changing in real time, depending on what the functional correlate is that's being studied at the same time. And I imagine that in a broader sense, that can then be kind of being integrated into other types of approaches like neuroimaging approaches that perhaps provide a more of a broad picture. No, actually, I guess pun intended, a broader picture of the whole system as opposed to these more detailed and in vivo sampling kinds of ideas.
You could certainly customize ligands based on what you find. But the other thing that I think is really timely is that the proteomic approaches, and I'm particular to one vendor, but I'm not going to mention the vendor's name, but using very small volumes of fluid like cerebrospinal fluid, you can measure the levels of thousands of proteins now. And it's expensive to do, but it's scientifically worthwhile if you have the wherewithal to do it. So one of the experiments we're proposing in our next round of studies is to sample the cerebrospinal fluid and sample the blood at the same time points. And then you can measure thousands of proteins in both spots. And you'll see which proteins that show up in the cerebrospinal fluid show up in the blood. And thus, give you a better biomarker. You can imagine doing this with Alzheimer's patients, and people have. You take cerebrospinal fluid from a few hundred Alzheimer's patients, and you take blood from them, and you do proteomics in both of the samples. What are the real markers that relate to cognitive status that are in the cerebrospinal fluid, and which of those show up in the bloodstream? And you could use such a powerful approach to look at biomarkers, accelerate the development of new biomarkers.
Yeah, that's fantastic. I think I've heard recently that hopefully there's going to be a blood test that's going to be available fairly soon in terms of early stage Alzheimer's diagnostics, but it would be interesting then to continue on in that vein as you're describing, and perhaps get even more specific tests around perhaps looking at being able to predict whether or not someone is likely to improve on a cholinesterase inhibitor. Because right now they have this, as I'm sure you're aware, typically this rule of thirds that they talk about clinically, that maybe a third of people will do well on a cholinesterase inhibitor, a third of people it's equivocal, and a third of people probably don't gain much benefit. So being able to anticipate that ahead of time with these kinds of more targeted biomarker tests would be really helpful.
Yeah, and Ben, they don't know if it's the metabolism rate of the cholinesterase inhibitor, for its ability to elicit action varies by person either. Obviously, since we're studying acetylcholine, we've actually used those in our lab animals and gotten similar cognitive effects in the short term anyway.
Mm-hmm. Yeah, I was interested because I can recall when I was at that poster session and talking to your colleague there, Kendall Pennington, that this particular study did involve some comparison with donepezil. Do you mind maybe helping us understand how that works?
Right. So the particular study, and again, it's published in Brain Stimulation this past January with Kendall Pennington as first author. We provided deep brain stimulation to macaque monkeys for about an hour a day, and we measured their working memory duration using a two alternative forced choice delayed match to sample. And we set up the behavioral tasks so that it would automatically get harder if the animal got a few trials in a row correct, and easier if the animal got some wrong. So it would automatically adjust to the animal. The animal would choose its own task difficulty. And by simply recording the task difficulty the animal worked through, we can see what the animal's working memory duration was. And so over a 12-week period, they got stimulated for an hour a day. The animals got, on average, 50% increased duration of their working memory. And that was then sustained for a period of time after that without further stimulation. The donepezil comparison, we took separate groups of animals and simply gave them donepezil each day and had them do the same task. But because donepezil causes an acute effect in the task, we gave them the donepezil right after the task. So the donepezil would be loaded into a date, which monkeys like to eat dates. And so the monkey would do his behavior for the day, and then he'd get a date as his reward, which had the cholinesterase inhibitor donepezil in it. And we used as high a dose of donepezil as we could get, because the animals were doing appetitive training, so they'd work for little food pellets that they liked. But if you give them too much donepezil, they don't really like food as much anymore. So we had to back off on some of the monkeys, but we gave all of them the most that we thought they could tolerate. And one of those 5 animals had a pretty pronounced improvement, but the other 4, we didn't see anything, which matches the rule of thirds with donepezil. And the clinical data too, that a small fraction of patients seem to do quite well with donepezil, and most of them, it seems like it's not doing anything.
Yeah, that's interesting for sure. And so those 4, what I was recalling from reading the poster was that, and then the other thing I was going to mention for viewers and listeners is I'm going to put links in the show notes to the relevant papers and to Dr. Blake's lab. So that I would encourage people to check that information out. But then, so then just going back to the results in that study, so the 4 that were help me understand that the 4 that were stimulated showed the improved working memory duration.
Yeah, we measured their working memory in terms of duration because the task automatically adjusts for 5% correct. So they all got the same percent correct, but they worked up to harder levels after they started to receive stimulation. And we measured that by the time between the sample and the potential matching stimulus in the delayed match to sample. If they're matching a colored square. The colored square might be yellow as the sample. The screen goes blank for 5 seconds and they have to choose between yellow and blue. And they choose the yellow, they get a food reward. After we started stimulating, they would do what they used to do at 5 seconds. They could do after 9 seconds or 9.5 seconds. It's about the size of the effect.
I see. And so the stimulation was in that, as you said, the nucleus basalis of Maynard. Is that correct?
Yes.
Okay. Huh.
We
So I
target
guess,
it, and for those who aren't, and most people don't know where the nucleus basalis of Maynard is off the top of their heads. Even I gave neurology grand rounds. Most of them don't even know off the top of their heads. But I think the easy way to explain it to people is that it's a sheet of neurons, and you could argue whether it's distinct from the globus pallidus or not, but it's at the floor of the globus pallidus or slightly below, and it's contiguous with the globus pallidus structure. So if you can find the globus pallidus right underneath it, you'll find the nucleus basalis of Maynard.
yeah. And then I guess, again, for folks maybe who don't have the necessary sort of background, then my understanding is that's basically the nucleus of neuronal cell bodies then that project forward with those cholinergic pathways that seems to be really important in terms of not only pathology of neurocognitive disorders like Alzheimer's in particular, but important as far as therapeutic targets with the cholinesterase inhibitor class of medications.
I usually explain to people that there are projection neurons there in the nucleus basalis of Maynard. Over 90% of those projection neurons release acetylcholine, and that they're most active during periods of high alertness and arousal. And so you can think of it as almost circadian rhythm driven. You're going to have higher rates occurring in the middle of the day than you will close to the ends of the day. Very little during slow wave sleep. They're paradoxically quite active during REM sleep also. But so we go into that structure, and in these animals, we max it out for about an hour. We just take it to the highest level of acetylcholine release that we can. And it took us quite a while to figure out how to do that. But that's the basic gist of the idea is we give their brain a big alertness and arousal workout for about an hour each day via the electrical stimulation.
Yeah. And I guess that's interesting too, because I think conceivably these kinds of results could have broader implications outside of neurodegenerative disorders and into things like potentially shorter term neurocognitive disorders like delirium, or even earlier life neurocognitive disorders like attention deficit disorder.
Yeah, I think that there's actually a large number of applications we think about and talk about in the lab. I'm pushing into Alzheimer's first because I think it's the greatest need is there, and I think it's got the greatest chance of working there. But yeah, if it has the positive effects in some application, then all the other applications become much more explorable.
Mm-hmm. And then, I guess also in terms of that existing patient population where deep brain stimulation already has a presence, say for Parkinson's, because obviously with Parkinson's, there's a cognitive component later on in the illness generally. So you could imagine that there may be, again, neurosurgical colleagues that might be interested in exploring additional targets for deep brain stimulation in that kind of patient population, also not only to improve the movement disorder, but potentially also to improve the cognitive component.
So that's been done actually 4 or 5 times already. Joachim Freund in Cologne, Germany, led the studies of this 15, 16 years ago, and with Volker Sturm as the neurosurgeon, and they applied stimulation nucleus basalis of Maynard in Parkinson's patients, as you note, because they're already getting deep brain stimulation. So they put in an extra lead and stimulate the nucleus basalis of Maynard, but they used a different stimulation pattern. And the same thing happened in several other trials in Parkinson's patients and Lewy body dementia patients. And in fact, they ran it in Alzheimer's patients, always using the same pattern of stimulation that Joachim Freund had developed. We use a different pattern of stimulation, we get different results. So I noticed that a new group from the University of Toronto, led by Alfonso Fasano as the PI, the neurologist, and his fellow Sanskriti was the point person for the study. They took 6 patients with Parkinson's that were getting GPI stimulation, globus pallidus stimulation, and they inserted the lead in a way that allowed the lowest contact on the lead to be in the nucleus basalis of Maynard. And they applied the same stimulation that had been developed by Joachim Freund, this 20 pulses per second stimulation, continuous pulse trains. And they didn't see a long-term positive cognitive benefit. And so, as it turns out, this is one of those strange terms, right? One of my friends emailed me their paper and said, how is what you're doing different from what they're doing? I read their paper and I said, wow, I might be able to help these people. So I sent them an email. And I said, hey, we study the same things. I'd love to talk to you guys and show you what we're doing. We always see positive cognitive benefits. And so we had a Zoom session, and by the end of the Zoom session, Fasana was instructing Sanskriti to go consent all their subjects that already had the electrodes in place. And so they did that, and they got a very nice cognitive effect, which was published in about a year and a half ago, also in Brain Stimulation, in a letter of update to their prior manuscript that did not show positive cognitive change. So the pattern of stimulation does matter, and you've got to get it right. And it did help cognition in those Parkinson's patients. But I think that the quality of life for a Parkinson's patient at the point where they're getting the DBS leads, if you improve their executive function a little, it's positive. It's something. I want to say it's not worth doing. It's definitely worth doing. But if you could prevent an Alzheimer's patient from losing their activities of daily living, I think that would be a far larger impact than altering the executive function in a Parkinson's patient who was already at the stage where they're getting deep brain stimulation. In any case, I'm collaborating with a group at Stanford and Helen Bronte Stewart's group. She is taking patients who are getting STN stimulation for movement disorders, Parkinson's patients, and they're also getting a second lead in each hemisphere to stimulate the nucleus basalis of Maynard. They think that they can improve on the stimulation we developed using tractography to find the right white matter tracks and put the stimulating lead there. I told them you should just copy what we're doing to get started with, and then do whatever you want. Just see if what we do translates first. And so they're actually doing both, and they're starting to implant subjects right around now. I was told it was going to be last month, May. I haven't heard from them. We have an update in a few weeks, so I'll know more then. But there should be stuff coming out of Stanford, and it's an open label trial. So that'll be reported at neurology conferences in the next year or so.
Yeah. Wow. That's fantastic. Yeah. We were talking a little bit about, just before the recording started, about how there's just been such a dramatic acceleration in terms of all of this research and the associated clinical implications over the past 5-10 years. It's really fascinating.
Yeah, I think two things really strike my mind right now as just absolutely exploding. One of them is vagal neurostimulation, and the other is the SAINT protocol from Stanford, the transcranial magnetic stimulation for depression. Such pronounced positive effects and just growing like crazy.
Yeah. No, it's fantastic. The other thing is that with the more of the invasive stuff, it's obviously there's a lot of the hype and the hyperbole, maybe, or maybe not, with these companies like Neuralink talking about implantation and all the things that they're working on. I think it's obviously it's great that there are folks who are benefiting in terms of the motor function recovery that they've been showcasing. But I think, doing all of this careful and very diligent sort of groundwork, including the basic science, and just making sure that obviously when it comes to invasive kinds of interventions, there's a huge safety aspect to it that has to be very carefully researched in terms of making sure that when it gets to the point of being clinically available, that it's going to be safe for people to have this kind of thing done.
Yeah, no, I think there is a huge threshold burden to cross to be able to do what we're trying to do. And there should be. We're talking about inserting a device inside somebody's cranium. There's a neurocritical care recovery associated with that. And that's not something anyone should take lightly. But at the same time, I think there's a really good chance that we can back up the cognitive aging clock in somebody with Alzheimer's more than 2 years and give them a few more years in which they have their activities of daily living intact. And I think that would be well worth the investment. But we've got to get there first, one step at a time.
Yeah, for sure. There's a huge clinical need. I know from my own practice, and we were saying the majority of folks really don't improve much with cholinesterase inhibitors. And I think from a pharmaceutical perspective, there doesn't really seem to be anything on the horizon that's promising around certainly disease modifying, but even symptomatic treatments for Alzheimer's. So I think that's fantastic what your vision is. I think there's a huge clinical need for that.
Yeah, no, I've talked to several people who have been really active in developing therapeutics for Alzheimer's, and they both said the same thing, that there's no sign of anything that has a positive effect on executive function. Everything is designed to slow the degradation. And we'll see. We've got to do clinical trials. It's a long road from here to there. And even if it works, there's 6 million, 7 million Alzheimer's patients in the US right now, about 1 million new patients each year. And there's about 150 functional neurosurgeons. And it doesn't take very much math to see that getting 150 neurosurgeons to implant 1 million people a year is a daunting task.
Yeah. I'm just curious, going back before we leave the nitty gritty of the study, one of the things I was curious about that you mentioned that you measured the changes in things like BDNF and NGF. So I'm just curious, can you talk a little bit more about that? Then what would be the implications around the changes that you found and how those are the implications around the impact that the brain stimulation had on that, as you say, the proteomics or the genomic implications of changes in the levels of those factors?
So nerve growth factor is part of the axon pathfinding process for cholinergic axons. And cholinergic axons in the cerebral cortex are responsible for things like angiogenesis. And so the thickness of the cortex, the blood supply to the cortex, all of those things are being boosted when you have larger amounts of nerve growth factor activating its receptor. And so we would expect to see the cholinergic axon should be branching more and releasing more acetylcholine, and the cortex should get thicker and there should be more blood vessels in the cortex. Those should all happen as a result of the stimulation. BDNF is a little more promiscuous. It has less selectivity for the specific cell types of which it's activating, but the most prominent are the parvalbumin sensitive or parvalbumin expressing inhibitory interneurons that synapse on the pyramidal neurons. And so that's a large part of the cortical circuit, primary inhibitory neurons synapsing on the excitatory neurons. So you'll have a stronger, more numerous connectivity between those types of neurons in the cerebral cortex as well as a result of our stimulation. What we observe practically is that the animal's working memory gets better. The monkey's working memory gets better. And the mouse, we saw better spatial learning in the Morse water maze. The magnitude of the effect, I think, is something that I don't speak about enough and I need to speak about more. We fine-tuned the stimulation in that paper published in Brain Stimulation earlier this year. And we found that we could reasonably expect to increase working memory duration by about 90% in these monkeys that are 25 to 30 years old, the equivalent of 75 to 90-year-old humans. So a 90% increase in working memory in a monkey is equal to about 1/3 of the lifespan of age-related cognitive decline in working memory duration. So we're taking 30 years off of their cognitive brain age by applying our stimulation for a few weeks. And that magnitude of effect is why I'm really positive about why I would even think about going into human studies and suggesting somebody get an elective neurosurgery, which sounds like you gotta have a pretty high bar to pass to be willing to do that. But I'm confident that we can pass that bar.
Yeah, that's fantastic. I think it really helps to put it in those terms for people to understand. That's really interesting. Yeah, it's really exciting.
We'll say one step at a time.
Yeah, for sure.
To the FDA.
Yeah, that's the thing. Yeah. So maybe that could lead into some discussion about how, I guess, in a broader context, how do you see that kind of work potentially fitting in terms of the broader landscape of that interface between neuromodulation and aging, particularly cognitive function, but aging research in general?
So right now, there's not much of a footprint of neuromodulation in aging research outside of obviously for movement disorders, but not for executive function treatment. And there's a huge need. And so if we're successful, obviously it would take off, but as I said, it's going to be limited by the number of functional neurosurgeons that are out there. But it can also recenter research. More people are really focused on how we can boost the function of the basal forebrain because it has positive effects on executive function in aged individuals. Then there can be other treatments that develop that have less burden on the patient and are more scalable. I think that's the worst case scenario for if we go in humans and it works, and then we have this problem. We can only do it for so many people each year, but I think other people will notice and they'll start to study the same processes.
Yeah, well, that's interesting. So what kinds of projects is your lab focusing on now in terms of continuing on in this path or sort of future considerations?
Yeah, so we have a mouse lab where we use the neuromodulator sensors, GATCH 3.0 or GRAB-NA 2.0. These are molecules that insert into cell membranes and they change their fluorescence when they bind a neuromodulator. In the case of the acetylcholine one, So we inject a viral vector into the cerebral cortex, and then we can image changes in acetylcholine in the cerebral cortex of the awake mouse. We do this in combination with the deep brain stimulation electrode, and then we have a direct readout of the neuromodulatory release caused by our stimulation. One of the interesting things about our stimulation is that it has to be applied in an intermittent pattern. You need to apply a large number of stimulation pulses in about 10 seconds, and then you need to wait for recovery, and then you need to apply a 10-second period with a large number of pulses again. We can watch this happen in the mouse preparation using the fluorescence sensor, and we can see the fluorescence go up and down in timing with the stimulation pulses that we're delivering. And we can use that to fine tune the stimulation in ways that we can't really do when we're only assessing cognition in monkeys or just guessing otherwise. Because in movement disorders, when they start stimulating a patient, they have them hold their hand up and the hand's shaking, and then the hand stops shaking when they start stimulating and they're in the right spot. We don't have that kind of instant feedback for executive function. And so we're seeking to get something that's more directly measurable. So we have stuff going on in the mouse lab to do that. The monkey project, we're looking at more proteomic approach of measuring cerebrospinal fluid centered around deep brain stimulation, also centered around doing executive function behaviors. The same approach can be used. We can draw cerebrospinal fluid before, during, and after the monkey does an hour of working memory behavior and see what changes the brain normally encounters when you just think really hard for about an hour. And we may add exercise to that as well. And on the human side, I'm pushing through to get the first human trial paperwork in order. So I'm talking to the FDA this week, and I'll be talking to the IRB probably next week. And then there's a process, and hopefully by the end of the year, we'll have our first subject implanted, and we'll be stimulating and trying to see to what extent these effects translate.
Yeah. Wow. That's so exciting. And I think it's really important to emphasize that it's so interesting how you're describing by the animal studies, these real-time, the investigation of the real-time changes, as you say, in the fluorescence kinds of studies in the mice. And then it's also really interesting to think about how you're actually measuring the real-time changes in things like cognitive performance in the monkey studies during the stimulation. And in particular is interesting in terms of thinking about how measurement of those real-time changes in executive function, for example, might reflect some sort of like an additive effect that is partially dependent on the nature of the cognitive task that the animal's actually undertaking while the stimulation is sort of there. Maybe there's almost like a synergistic effect that wouldn't be there. It's not as if the neurostimulation is creating I don't know, it's more of a boosting or perhaps like a potentiation kind of effect. Is that what your team is finding?
I really want to apply stimulation in a human and ask them, because
Sure.
you can't ask the monkeys.
No,
But
right.
I
I
think
guess.
it's all reasonably consistent with them having a heightened level of alertness and arousal. We all go through daily lives, and some hours we're sharper and more productive. And then sometimes you get to the late, especially me at my age, get to the late afternoon and it's, wow, I can't do that stuff I need to do. I'll do some stuff that has less cognitive demand for the next couple hours. And so maybe we're just elevating all of that and that ability to be alert and aroused for longer periods of time. So I'll love to find out, and hopefully I'll get to do so.
Yeah. It makes me think it would be interesting to potentially look at an additional factor as far as a medication like modafinil or something that, could potentially even enhance that even more, or even looking at the implications of medications that are known to block that kind of thing, like an anticholinergic type agent, see how the deep brain stimulation may or may not counteract that. And so the interface again between the pharmacologic factors could be an interesting approach as well to look at.
Yeah, and we've done some of those studies in the monkey with the short-term effects. It's much easier to do that sort of study where you're looking at effects that happen within 2 minutes or so, because you can go through different conditions faster. When you're looking at something that takes weeks to accrue, which is what the cognitive effects of the deep brain stimulation are right now, it becomes a lot more, you have to choose your studies more carefully, create a priority list, and you don't get to do very many of them, because once the monkeys get smarter, it's not so facile to make them go back to where they started.
Yeah. I'm curious. I think that there's, like I was saying just now, there's been a lot of hype in the press about these brain computer interface initiatives and that. And on the one hand, I can just I'm obviously I'm a clinician primarily, and from that perspective, from the perspective of an interested member of the general public, it sounds interesting, but I could also imagine that from the perspective of a researcher who's got, decades of experience in this kind of field, that perhaps there's a little bit of skepticism with these high-tech kinds of approaches. So I'm just curious, what are some of your thoughts around that whole landscape in terms of this BCI tech world descending and innovating around this kind of thing?
Right. So my previous career, I designed and built cortical implants, and we used them to study sensory cortex, but other were doing the same work at the same time, looking at motor systems, and those became the early brain-computer interfaces. So I've been following the field. I know a lot of the primary investigators in that field. I think that there will be brain-computer interfaces that really help people. And I think the first one, and now I am going to mention a company name, is going to be Synchron. And what Synchron does is they have a stent that has contacts on it, and it gets inserted in the sagittal sinus, which for those who aren't familiar, is a blood vessel that's right on top of the brain. And they can insert it endovascularly. So it's an outpatient procedure. And the wiring for it then runs down through the jugular and crosses the wall of the jugular right around here. And they have a device that they can communicate with. So they place it in an outpatient procedure. The next day, the subject can control a cursor on a computer and can click. And in so doing, they can type words in 10 or 12 seconds. And they couldn't do that today. We're talking about somebody like an ALS trapped patient. And all of a sudden, they can tell their wife, I love you, for the first time in months. And it's just brilliant and inspiring to see. The function of it, it's not very high. It doesn't convey as much information as those Neuralink probes do, but it does convey enough information to allow the patient to type slowly. And it's very safe, and it's going to be very inexpensive, at least in the relative sense. There are others that I'm really positive about are the ones that are going to use the ECOG grids to generate a brain-computer interface. Now, an ECOG grid is a flexible plastic sheet that gets inserted on the surface of the brain. And the benefit of using the ECOG grid is that you get stable signals from most of your contacts for long periods of time. And you get a reasonable reflection. It's grossly average. You're not recording from single neurons, but you're getting an average over millimeters of cortical tissue each of what the cortical activity is. And people have shown that you could actually get a pretty good brain-machine interface if you have an ECOG grid that has enough contacts on it. But even if you only have a few contacts on it, you can probably get a highly functional brain-computer interface. And again, it's going to be low cost and low burden on the patient. The Neuralink approach, which because of who's behind it and budget and so forth, has gotten a lot more press. But I'm not convinced that the penetrating electrode approach is going to be that useful. And I remain to be convinced. I'm skeptical because those were the probes that we developed 3 decades ago, and we could do the same things Neuralink could do then. But the problem is consistency from subject to subject and also consistency over time. And the ECOG grids and Synchron don't have those problems. They're consistent over time, and they're the same in every person. And those are beautiful things when you're trying to make a commercial device. If you're losing most of your contacts or most of your recording sites in the first month, but you still have some, but the yield from subject to subject can vary by an order of magnitude, which is what we always found with the penetrating electrodes. It becomes much harder to turn that into a commercial reality. I think that just at the planning stages, the ECOG grids and the Synchron approach, the stent and the sagittal sinus, those are really going to help people. And I think they'll both be FDA approved in just a few years.
Yeah, now that's very interesting. I have definitely heard of Synchron, and I think what you're saying makes total sense, partly because, as you alluded to a couple times earlier, is that I think a rate limiting step is probably going to be access to the neurosurgeons or the interventional neurologists. I guess maybe that's going to become an increasing clinical area, is the limiting factor for accessibility and commercial sort of scaling.
Yeah, I don't know if you've been in the neurosurgical and neurological training units much lately, but we are sure training an large number of interventional neurologists these days. It seems like a lot of our neurosurgical residents want to do that, but also a lot of our neurology residents want to do it. They say unequivocally, you can help a stroke patient, for example, in ways that are really almost impossible to do otherwise. It's really the biggest advance in stroke treatment in decades. And on top of that, there's new things coming along like the Synchron stent that would be put in endovascularly as well. And so I think there's every reason to think there's a big future for those folks in that practice.
Yeah, that's super. That's really exciting for sure. I guess maybe wrapping up, I'm curious to know in terms of your career in providing mentorship and training grad students and postdoc fellows in your lab, for any of those such folks that are watching, listening, trainees, early career neuroscientists interested in this kind of translational neuromodulation research, is there any thoughts or advice that you might give in general terms about choosing, like we talked about at the very beginning, choosing impactful research questions or pursuing specific research ideas?
Yeah, I always advise trainees to work in areas that are growing and not in areas that are shrinking. That's just seems you could make that same advice if somebody was working in business, sales, or just about anything. You want to work in something that's going to be more desirable 10 years from now than it is now. And so most of neuromodulation fits that. I would think deep brain stimulation does, and also vagal nerve stimulation and also non-invasive stimulation. They're all growing fast. And so those are all good things. There's really a critical shortage of people with biomedical engineering training that look at the biological interface and the effects on the biological interface. And I say there's a critical shortage because all five of us know each other. There's more than five of us, but there's far fewer than you might think, and we all know each other. And that excludes most of the clinicians who don't really have such a grasp of what the stimulation pulses are doing with respect to the biophysics. How do you make a contact that will work? What metals do you use? How much current can you push? What is the effect of the pulse? What happens when you change the pulse width? What is the volume of tissue activated? And how do the fields that are caused by transcranial magnetic stimulation, how are they the same or different from those you would do with deep brain stimulation? And so I think that those are areas really to go into if you're going to biomedical engineering, and Duke has good people, Case Western has good people that train in those areas. And if you went to one of those two and you've worked on those, you would have a firm job in neuromodulation ad infinitum. The more neuroscience oriented people, I think I really like working at least part in humans, because that's where the rubber hits the road. And that's where you feel like you can really make the world a better place. And if you can do that and do some animal work, which is what I've been trying to do for the last decade, that's where I want to be, because I can both feel where the rubber hits the road and also see what's going on under the hood.
Mm-hmm. Yeah, no, I think thanks for that, offering that wisdom. I think that's really inspiring. And in general, thanks again for a really interesting conversation. And I think just with the sentiment that you just offered, I think it's fantastic. And just want to congratulate you and your team on your research to date, and I wish you all the best with your future endeavors. And yeah, I think it's great. I just think that the more that we can all collaborate in the interest of helping people and reducing the burden of disease that people are facing, so much the better. So thank you again. And yeah, just really appreciate your time and your expertise. And I think it's just going to be really valuable for folks to watch and listen to.
Thank you. And thanks for having this podcast. These sorts of things are part of the new media, and getting the word out there is something we all wish we could do more of. Thank you.
Fantastic. Yeah. No, that's great. I'm glad to be a part of it. It's just growing, but hopefully the more conversations like this that I can have and put out there, then the more interest it'll generate. And then we can, as you'd say, start to spread the word. Yeah. Appreciate your time, and all the best. Thanks again.
Okay. Thank you.
All right.
Thanks for joining us today on the Neurostimulation podcast. A huge thank you to Dr. David Blake for sharing his team's groundbreaking work at the intersection of deep brain stimulation, neuroplasticity, and cognitive function. If you would like to find out more about Dr. Blake's work, please visit his lab's website at Augusta University using the links in the show notes. You'll find everything that you need to dive deeper into this topic. And please join the conversation. I would love to hear your thoughts. Please leave questions or comments in the comment section below. Your questions, ideas, and feedback make this podcast better. And as always, I would encourage you to like and subscribe to this podcast. Share it with someone that you think might be interested. I really appreciate your attention, your time, and your interest. Until next time, be well, stay curious, and I'll see you again on the Neurostimulation podcast.